This is a production of Cornell University.
Hello, my name is Silene DeCiucies, and today I will be talking to you about the mechanisms behind negative priming of soil organic carbon by pyrogenic organic matter. So first, we're going to start off with the why. We're really changing scales and topics here from our last talk. We're getting a lot smaller, and so why are we concerned about these interactions? And the number one why is that pyrogenic organic matter is everywhere. So in recent history, it's been touted as this exciting new agricultural amendment, this magic bullet, but it's also been added to our soil for hundreds of years, or thousands of years, throughout human history, through cook fires, burn pits, and more importantly, it's been added to the soil through natural events, like forest fires, lightning strikes, for hundreds of thousands of years. So this stuff is ubiquitous in soils. And we now know that it has profound interactions between itself and the soil organic carbon that's already there. So it's impacting the soil organic carbon. And in recent work in our lab, we have found it's been found that this could have long-term implications for carbon storage. So this is all very exciting. The carbon is there, it's interacting with the carbon that we know and care about. And today we're going to kind of pull apart these interactions and break them down to a mechanistic level. So priming is a word I'm going to be using a lot today, and it's used differently throughout the literature. But in relation to my work, the priming of soil organic carbon by PyOM is basically describing a change in mineralization rate. So when you add PyOM to soil organic carbon, it might increase the mineralization rate. That would be positive priming. Or it may decrease the mineralization rate, which would be negative priming. And today, we're only going to talk about negative priming because this is what I've found in my research, and this is also kind of what's exciting in terms of long-term carbon storage. So this is a list of proposed mechanisms of negative priming, kind of gotten from current literature surrounding this work. And we're going to walk through them one by one. And these little icons, these colorful icons on the right, are going to repeat throughout the presentation today. So you'll hear these words a lot. You'll see these colors. When you see them in the upper right of the screen, that kind of means that whatever I'm talking about is related to that mechanism. So this is our baseline situation over here. We have soil microbes. They're eating soil carbon. They're respiring CO2. So for the first negative priming mechanism, we have inhibition of nutrient mining. And what's happening here is when you add the PyOM, it basically creates a nutrient source for these microbes. And so instead of getting their nutrients from the soil and respiring that carbon, they're going to get the nutrients from the PyOM. And that'll decrease their soil carbon respiration. So next we have toxicity. So this is pretty simple. Maybe you're adding the PyOM and it's killing the microbes, so it's reducing their respiration. All right, next, substrate switching. So we're adding a new substrate that the microbes might find more tasty, and so they're not going to respire as much soil carbon, they're going to respire char carbon. Substrate dilution, so this is kind of a little bit of the substrate switching and also a physical dilution of the soil organic carbon. So basically the PyOM addition is making it harder for those microbes to get at their tasty soil carbon. SOC sorption, so PyOM has a really high kind of adsorptive potential. It has high surface area, charge, and so what may be happening is that the DOC in the soil, or in the soil carbon in general, is being sorbed onto the surface of the char, and this is protecting it from being mineralized by microbes. And then microbial signal sorption, so this is something I'm not really going to touch on today because I haven't worked with it at all. We're doing some of that in our lab, but it's exciting stuff, and to break it down to its simplest form, essentially perhaps PyOM is creating a barrier between the communication of microbes. Say one of them is saying, hey, there's a bunch of tasty food over here, and the PyOM is blocking that from getting to their fellow microbes. So it's going to reduce the amount that they're respiring that carbon. And then the last one is kind of facilitated aggregation, in that when you add PyOM, perhaps it's increasing the ability of that soil to make aggregates and cause physical protection. So these are kind of the mechanisms that are in the literature right now. and that we'll be kind of talking about today. So overall, my overarching question is which mechanisms are responsible of that big list that we just talked about? And today I'm going to break this into a couple different questions and topics. First, we're going to talk about the dilution mechanism, followed by substrate switching, toxicity, and then we're going to see kind of what's left after that and how might we parse that out once we've gotten rid of a couple of these. And so our hypotheses, as I was mentioning, we know that PIOM is very adsorptive, so we're thinking that sorption is a really important mechanism, but we also know that there's probably more than one going on, and so we're going at that with this approach, kind of keeping an open mind throughout this mechanistic study. So to look at the dilution experiment, I set up an experiment where I'm basically trying to simulate the dilution effect with other inert media. So I'm adding PIOM, and then I'm adding these different media in the same volume equivalent. So I've added perlite, quartz sand, and also a carbonless soil version of the soil that I'm using. In this experiment, I use Marden topsoil from Mount Pleasant. That's about 5% carbon. And I'm pH adjusting the char so that it equals that of the soil. And I'm also adding nutrients that kind of overwhelm any nutrient effects that you would get from adding the char. So this experiment is really targeting the mechanisms of dilution, but also substrate switching. And because of the way that we've created the experiment, we're able to cross off a couple mechanisms. So we can get rid of nutrient mining because we're adding more nutrients than the microbes could ever really need, or that they could get from that extra char. We're also able to cross off pH effects because we're pH adjusting the char so that it equals that of the soil. And then also, this is a pretty short scale incubation. It's only about 30 days. And so we can kind of cross off aggregation because that's not quite enough time to really facilitate those type of interactions in the soil. So this slide is kind of walking through my general method of investigation. This has simplified basically like 2 years of my life in one slide. So what we did is I grew isotopically labeled willow biomass. This has a really super high 13C label. And then we're creating PIOM out of this biomass. So for this experiment, I'm using a 450-degree char. And then we're adding that char to the soil and also our other amendments or other treatments. We're wetting these, and then we're monitoring their respiration over time. And we monitor the respiration using this fancy machine on the 7th floor, the Picaro. And essentially what this thing spits out is a ppm CO2 respired per day, and also an isotope value that corresponds with that CO2. So with those two numbers, we're able to throw a little bit of algebra at it and basically determine how much of that respiration is coming from the soil and how much is coming from the char. And through that calculation, we're able to determine priming. So these are some results from that first experiment. So we can see, first I'm going to explain this chart actually because I'm going to be using these units a lot today. So on the y-axis, we have cumulative soil C respired divided by the initial soil in the jar. So this is normalizing every treatment based on the amount of carbon that they started with. And it's also cumulative, so it's adding the amount of soil carbon respired day by day. So what we see here is the black line is the control. So that's just the soil. We've added nothing to it. And in this particular experiment, we experienced negative priming for all of our treatments. So even just adding sand, completely inert, really low surface area media, we had some negative priming with that. But we can also see that the perlite, maybe a little more surface area, more negative priming, followed by this ash subsoil, contains clay, probably a lot more interaction going on with that media. And then finally, we have the most negative priming with our PIOM addition. And so what we can say about dilution is that perhaps the dilution effect can be explained by this difference between the control and the sand, or even the control and the perlite. But this isn't completely compensating for the amount of negative priming that we have from our char. And so this is another way to look at this data. So this is, the same treatments, but instead of cumulative, it's looking at daily soil carbon respiration. And so you can see that most of this effect, most of this negative priming is happening rather quickly within the first 5 days. And also on this chart, I've added the respiration of the char itself. So you can see that this is pretty negligible compared to the soil carbon that's being respired. And finally, what I've done is I've taken each treatment away from the control so that we can really have a look at the magnitude of priming that's going on. So the control doesn't exist in this experiment because it's 0. That's what this line is right here. And what we can see is this increasing negative priming, kind of weird language when less is more. But we can see that, yes, we have significant negative priming with this PIOM treatment. And up here is the respired char. And so we can also see that the respiration of the char definitely can't compensate for the amount of negative priming that we have of soil carbon respiration. So overall, from that experiment, what we can say is that dilution definitely isn't the only story. Neither is substrate switching, but we're going to get back to that. But both are definitely having an effect. They're exerting negative priming. And this is kind of showing us, of all this negative priming exerted by the char, about this much at this point can be described by this dilution effect, and this much by substrate switching. So we still have this huge part of the pie that's unexplained mechanistically so far. Okay, so now moving on, we're going to target substrate switching as well as soil organic carbon adsorption. And so the next questions to get at these new mechanisms, now that we've kind of crossed off dilution, is how can we determine, and also toxicity. So that's an important one. So first, for toxicity, how can we determine that the char we're applying is not just killing these microbes, and that's what's causing these interesting effects? Next, can we make a better case for refuting the substrate switching that might be happening? And finally, if adsorption is this driving mechanism, kind of does it have an endpoint? Is there a smaller amount of char we could add that wouldn't have this exciting negative priming? So to answer these questions, I designed an application rate experiment. And this is really simple. It's just adding kind of different concentrations of char to the same soil that I used in my last experiment. So we're adding 0.5, 1, 2.5, 5, and 10 milligrams per gram char to the dry soil, that same 5% carbon soil. And we're trying to see if there's an ideal application rate for negative priming, and also if negative priming doesn't exist at a lower application rate, because maybe all the adsorption sites of the char are totally used up. And so other factors are overwhelming this negative priming. So we're kind of trying to see if it has an endpoint. And this is kind of a simplified version of the results, but it's the same type of figure we saw before, cumulative soil C respired. And here we have our control again in black, but we have positive priming as opposed to the last experiment where we only had negative priming. So when you add the 1 milligram per gram application rate, we're actually increasing the rate of soil organic carbon mineralization. But when we have the higher application rates of 5 and 10, this is decreasing the mineralization. So it's important to note that, yes, char is kind of causing negative priming, but it may not in all situations. And this is something we'll kind of have to parse out in the future. But maybe if it's a mechanism like adsorption, those sites are being used up. There's no more sites left to adsorb the carbon. So we have positive priming. And again, this is that same kind of figure, taking away each treatment from the control to see the magnitude of priming. See the positive priming over here, the negative here. And again, to speak to the substrate switching mechanism, which, if we can all recall, is where the microbes are eating the char instead of the soil. This, basically, the accumulation of the soil, of the char carbon respired would describe that substrate switching. And we can see that the magnitude of this respiration is nowhere near the magnitude of our total negative priming. So we can really kind of cross off substrate switching at this place. And to speak to toxicity, this is kind of the easiest way that I could think of to kind of assay how microbes are doing after being exposed to char. So this is a fumigation extraction method of microbial biomass. So after I've incubated my char in my soil for 40 days, I completed this assay. And what this is saying, this is kind of a messy graph with huge error bars, is basically that there's no significant differences in microbial biomass communities if we're adding char or PIOM or if we're not. And this is looking at both a topsoil and also a subsoil, so a higher carbon soil and a lower carbon soil. So we can kind of say from this that toxicity isn't really occurring, which is what this is saying over here. And so now we're going to move on to what we have left. So finally, we're going to talk about SOC sorption, because that seems most likely as a candidate for our negative priming that we're seeing. And a really neat way to look at sorption is just creating these adsorption isotherms. So I've taken different concentrations of DOC from the soil I used in my incubations, and I've shaken them overnight with different media. So either a 200-degree char, 450-degree char, 750-degree char, or these two soils. And what you're seeing here is over here we have desorption. So below the 0 line, all this DOC was desorbed. So we're seeing complete desorption for this this low temperature char, as well as the topsoil, and kind of the subsoil is right in the middle. And then for our chars, for our higher temperature chars, we have desorption at lower concentrations, adsorption at higher concentrations for the 450-degree char, which is the one I used in the experiments I've talked about already. And we have almost complete adsorption with a higher temperature char. So as you increase the temperature of pyrolysis, you're having more adsorption. Another really exciting way to look at adsorption in situ is nanosims. So I've labeled my char very highly with delta 13C. And using this amazing technique, we're able to look at the char and see what kind of carbon is sticking to it. So the char by itself, so this is one that's been unincubated, so it just has this super high delta 13C value, which is indicated by this purple color. And this is a piece of char that's been incubated with soil for 40 days. And so we can see that on the surface we have these mottled colors, we have a lot of this blue natural abundance carbon. So what this is telling us is that over time we are seeing adsorption of natural abundance carbon on the surface of char. This can't tell us what exactly we're looking at or why this is happening, but it's kind of a new, neat way to be able to look at sorption in situ. And hopefully in the future, this will be kind of more quantitative science that we'll be able to make more conclusive, more conclusions with. So in conclusion, basically, we've been able to cross off or minimize a couple of these mechanisms today. And so, and based on our results, we're thinking that SOC sorption is an important mechanism. We also have a lot further to go with things like microbial signal sorption, kind of aggregation. And we also know that this is probably a combination of many mechanisms. As we've seen today, we can kind of compensate for part of this pie, but there's so much left. And is all that really coming from sorption? Maybe, maybe not. We've basically ended the day with more questions than we started with. But it is called research for a reason. And it's exciting to keep moving forward. And this mechanistic approach is just, I really love how really deeply scientific it is. It's just kind of probing at the teeniest details and being able to build further from that. And I've really enjoyed this type of science. And it wouldn't be possible without my committee, Johannes and Janice, and also everyone in my lab who helped me out, especially our amazing research technicians, and really everyone. This couldn't have been done without my lab and without all of you. Thank you. And questions?
Yeah, thanks a lot. Very nice and clear. Can you say a little about how the soil was handled before, from the field to start the experiment?
Yeah.
And if I can throw in a part B, did you think about adding radiocarbon as kind of another?
Yeah. So the soil, I got it from Mount Pleasant and kind of homogenized, took out all the rocks and roots, and dried it out, sieved it, and then completely dried it down. And then I've messed around. I've done a lot of iterations of the incubation where I've pre-incubated the soil. But for these experiments today, I mixed them with the amendments dry and then added water right before it started, which I found was just kind of the tightest way to keep everything the same. It's really hard when you're mixing mud and char together. So that's how they were treated before. And yes, I have other experiments where I have added. Usually I'm adding an uncharred version of the PIOM, so I am adding biomass. And in those, we're basically seeing we are seeing negative priming, but it's mostly compensated for by this substrate switching. So you can see this huge surge in the microbes eating that like spicy, exciting carbon source, and it's definitely different mechanisms than with the char. Any other questions? Yeah.
So I also two questions. So one is, and I really like the mechanistic approach to everything. So one is the 5% organic carbon in that soil. So that was the way the soil was naturally.
Yeah. Yeah, it's kind of the top.
Was it from the forest? Or?
Yeah, it's kind of like the topsoil of Mount Pleasant.
Oh, okay.
Yeah, 4.5, 5.
The other thing is, so maybe I missed it a little bit. So you had to put your rates.
Mm-hmm.
Yeah, 1 milligram per 1 milligram per gram, right?
Yeah, I have 0.5, 1, 2.5, 5, and 10. And on my figure, I only showed a couple of them because the 0.5 and the 2.5 just kind of muddied it up. But essentially, the 0.5 exerted positive priming as well as the 1, as you saw, and the 2.5 exerted negative priming. But neither of those were different enough from the control. They just kind of made it look a little messy.
So what did, I was just kind of doing some calculations in my head of what, I mean, if you think about applying biotech to an acre of land, so...
Yeah, so for the...
like 20 tons, 10 tons, 2 tons, kind of...
So for the 5 milligram per gram application rate, which is the one that I had on the figure, so that would exert kind of significantly different negative priming, and that translates to about 4 tons per hectare. 4 tons per hectare, yes. No, 4 tons per acre, about 11 tons per hectare. Yeah. Mm-hmm.
And is that a lot, in terms of, I don't know what the bulk density is there, would that be a large
That would be, yeah, that would be a large application. So if you think about adding compost, a typical addition of compost is about 10 tons per hectare, 10 tons per acre, which is twice the amount of my application rate. But that has a similar bulk density, or even like much higher than the char, which is really fluffy stuff. So this would be like, yeah, we would be adding a significant amount of char. But it's also confusing because of the, the depth of soil and like that 8-inch plow layer. It's kind of really hard to translate it to this, but it would be like a significant addition of char. Yeah, it's not a sprinkle, yeah.
So in a way, it makes more sense than to apply a large amount at once than little amounts
over time. I mean, that's kind of what this is saying, but we definitely need to look into that more and break that down into, especially field experiments. I mean, this is in jars in a lab, so we need to extend this further, yeah. Mm-hmm. Yep.
I'm curious why you chose to simply remove the pH factor from the test. I mean, most of the work that's been done that shows negative priming, those are basically adding the char to the soil and looking at the results that way, as opposed to adjusting the pH. So
why?
I'm just curious why that ruled out that would.
Well, I don't think it should be ruled out, but I guess for me it wasn't as exciting to look at. So char has an inherently like super high pH, and a lot of the soils I'm looking at are around 4. So we know that's going to have a huge effect, and we know that that's probably, I mean, it's just kind of something that we know is happening, and that I just kind of wanted to look at different mechanisms. I wanted to try to control for that, and I know it's super hard to control for that because basically I'm adjusting the pH with HCl is how I'm adjusting the pH of the char, which maybe isn't completely, compensating for the pH changes. But for me, I was just kind of more interested in other mechanisms that we didn't know already had a profound effect. Like we know pH is so important. Like pH and moisture are arguably like the most important factors. But I was kind of trying to probe beyond those.
Yes, I'm looking at your pie chart, and you've got a very small amount, and then that big question.
Yeah.
The question, I guess, that I'm wondering about is what portion of that question is the pH effect, and what proportion of that is for these other two factors that you kind of hone in on, and they're still extremely narrow.
Yeah.
And then the rest is just pH.
It could very well be, and that's not something that I've looked at. So in the future, we should. Mm-hmm. Dr.
Let's put together the fact that your CO2 evolution measurements are for the whole soil, and your explanation of absorption as well. So the use of absorption as an explanation is based on absorption of VOC over on pyrolytic carbon, which in fact, if the pyrolytic carbon has a very high pH, the absorption of the mostly negatively charged VOC is going to be pretty much
Pretty much what?
reduced at high pH.
Reduced at high pH.
Yeah.
Oh, I don't know, because I've only looked at the system with these pH-adjusted situations. But, I mean, it would be pretty easy to do these adsorption isotherms with an un-pH-adjusted char to see what happens. Because as we're seeing in one of my first pieces of data, a lot of this negative priming can be happening very quickly. So within the scope of, a 24 or 36-hour adsorption experiment. So I guess we'll have to see. I'm not sure how the change in charge or pH will affect that.
Assuming that all the CO2 that evolves at the beginning in those first 5 days is all from the VOC extraction of the soil?
We don't know. It could be. I mean, that, adsorption is something that could be happening that quickly. And it would probably increase if you had a super high pH char. But that's not something I can speak to yet because I haven't tried it out. Mm-hmm. Yep.
I'm wondering whether there is a possibility that there is a changing about the composition of the microbial population with or without the term.
Oh, yeah, there definitely are changes. Thea, my kind of predecessor, did some work with that. Tara in my lab is also working with that. That's not something that I've looked at all. Kind of avoided it. Yeah. But anyway.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.